There we go. Get myself comfy. All right. Good morning, everybody. Happy Monday. Not too long to Thanksgiving, so. Okay, where are we at? All right, so hope you all did well in the exam. Had a quick look at the grades and looks like most people are doing actually pretty well, which is cool. And an improvement on exam one for most of you. So that's always encouraging to see. All right, so essentially what we have now for this section is we've got a little bit on eukaryotic genome structure and transposons. And transposons are actually really cool. Worked on those for a bit some years ago. And then really the rest of it is going to be all on gene expression, central dogma, well, sequence hypothesis really, and breaking that down into its individual pieces because it's a very complex process that is really, oh, here they come. What are you two going to do? I think they're still looking for the cat they were chasing around earlier. Yeah, little one is complete gonzo right now. I don't know what she's doing. Let's calm down, whatever you're doing. Anyway, get out of it. So, but gene expression is really, I mean, it's the central dogma, not so much because, you know, it's in the middle of anything, but it's central to everything biological, or really, yeah, anything living, to be honest, or even non-living, you know. And so it really deserves a good bit of exploration, I guess one could say. And also we're going to kind of leverage off of that and then we'll spend a good amount of time in the last section oh my God, these dogs. Looking at the control of gene expression, right? So it's not just turning the information in DNA into something useful. It's how you control that matters a great deal. These two are going to be super distracting. She does this kind of like World Wrestling Federation thing where she walks along the back of the couch and then jumps on our bigger dog. I don't know. It's funny to watch, kind of distracting to hear in the background as you're trying to work. We have 2 more exams. We have 1 exam on the section that we're going to be starting in just a moment, which is mostly about, gene expression. And then we have our last exam, which is not a final. It is another section exam, essentially. And that will be on control of gene expression and some, you know, biotechnology, transgenics type stuff, depending on how much time we have. So yeah, there'll be 2 more exams. The last exam is not a final, it's not cumulative. Yeah, I'll type this out, just is not cumulative in the conventional sense. It's just on the 4th section. But obviously everything that you've learned in the course to date or till that point will help you with that exam, right? So it's not cumulative, as in I'm not going to be quizzing you on stuff that we learned specifically in the prior sections, but what you did learn in those sections will help you understand the material in the last section, right? Because it is like the whole course in a sense is cumulative in that way, right? Hopefully that helps explain that a little bit. Okie doke. So let's move on. All right. So essentially what this is about is kind of putting together a bunch of stuff, basically. But it's really something that's important to. I should have had a coffee before class, not after it. It kind of ties together a lot of bits and pieces, essentially, and especially genome dynamics is a really important part of control of gene expression, as well as being like a physical solution to a problem. So it's important to get a good understanding of that. That's why we introduced it last section a little bit, and we'll be kind of riffing on it further over the next couple of sections. Hokey smoke. Anyway, so this is basically what, well, if someone took my chromosomes out and got them to condense, this is what my chromosomes would look like. Actually, this is from a dividing cell. The reason why I can tell is if you look really, really closely, you can see a very faint line down the middle of each chromosome, right? So that basically shows you the chromatids. As for the colors, I'm not entirely sure where the colors come from. They may be some immunohistochemistry, right, where they're labeled with antibodies against particular proteins that are associated with the DNA. Anyway, but like the whole point of this, we'll come back to it later, is that our chromosomes differ greatly in size, right? And they're actually called chromosome 1 to 22 based on their size, right? There's no other reason for it. And so they're pretty much just laid out really neatly, like poked around until, actually, I think they take pictures of them and then line the pictures up. It's a lot easier. Until they're in size, and that's how you tell which one is which, right? And you can also see this is obviously from a male because we have that nice little stunted Y chromosome down there. Okay, so when we talk about our genome is essentially our, when we talk about size anyway, that really refers to the number of bases or nucleotides, really, kind of depending on which word you prefer, in a haploid genome, right? So essentially, a genome consists of all of the units of information, right, that encode genes, right? Genes and other stuff, right? Actually, an awful lot of other stuff. So, sorry, I got woken up like really early by daughter and wife and then dogs. So I didn't get a very good night's sleep last night. So when we talk about the size of our genome, really, because we're diploid individuals, right, other than our germ cells, it's actually, we've got twice that amount of DNA. We don't have twice the amount of information, though, right? So our genome is really the unit of kind of information summed up by all of our individual chromosomes. This is what I've got going into my right ear while I'm trying to focus on genetics. It is a challenge. So as you can see, there's a pretty huge range in genome sizes, right? So actually plants tend to have the largest ones. I don't know what on earth Sarcodina are. What's Sarcodina? I'm a very poor taxonomist. In fact, I suck at taxonomy. It was one of the, oh, well, that's what they are. Oh, includes amoebas. There you go. Didn't know that. Yeah, actually, taxonomy or what was it? Insect taxonomy. I just flubbed it completely at university. basically I got to like the 7th group or whatever's of insects. I was like, this sucks. I'm going to do something else. And then the exam was on like groups 8 to 14. So anyway, I've got a bit of a long-held aversion to taxonomy. But forgetting all of that stuff, right? Essentially, we have, they're grouped in like type of thing, whatever they are. Fungi, animals, plants, and protists, right? So they're all eukaryotes. Single cell, typically protists are single cells. Fungi are, some of them are, some of them aren't, right? And so animals and plants are all multicellular. And here's us kind of right in the middle, more or less. But what you can see is that there's this huge range, right? Sponges are pretty tiny in terms of genome because they are super simple organisms, like really, really simple, really simple. But they're multicellular, but they're like super, super simple multicellular organisms. But then you got like mollusks, which aren't really that much more complex, have far larger genomes. In fact, amoebas have, some of them have the largest genomes that there exist in eukaryotes. In fact, what is it? Yeah, I was listening to a guy that I worked at where I was at last, who does a lot of deep sea biology, kind of barophiles or piezophiles, whatever you want to call them. And he said that on one of the last cruises that he was on the Google ship, can't remember what it's called, they actually found a amoeba that had a radioactive crust, right? So obviously they can get very close to it, even if they could. And it was about the size of a cantaloupe, however you pronounce it, small melon, right? An amoeba that big, right? Big as a baby's head, one cell, slowly going bloop, 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 along the bottom of, I don't know, whatever rocky shelf they were looking at. Absolutely crazy. Amoebas are pretty weird. So anyway, long story short, organismal complexity has nothing to do with genome size, right? And there's a range for even the same thing, like bony fishes, right? The ones that you have to kind of pick out as you eat them, right? Huge range from smaller genome than us to a far, far, far larger genome than us, right? And so, and actually, plants tend to have really big genomes, particularly because they undergo genome duplication. So actually, 2 good examples. I always like talking about these. Wheat, because, you know, I'm from the UK and wheat is like second only to potatoes, has a huge genome. It's actually hexaploid. And also the other one, leave her alone. Sorry, dog's chasing cat. Is maize, right? So what you use to make tortillas, right? So actually, if you drive around San Antonio, and particularly as you get further south towards towards Mexico, you'll see this kind of weird, like, yeah, kind of bushy plant at the side of the road. I actually see it sometimes on my commute, and it's called teosinte, right? And that actually is the ancestor of modern maize. And it got there through 2 rounds of genome duplication. So there you go. Anyway, and so this is basically called the C-value enigma or the C-value paradox, right? Either one. And the C-value is the DNA content of the haploid genome, right? So that's what this is essentially all about. And essentially it means that the size of the haploid genome size of an organism has nothing to do with its complexity or its evolutionary place in, you know, any kind of taxonomic or phylogenetic tree. Amor, you're gonna have to take these dogs away. They're just being absolutely batty. These dogs, batty. Yeah, crazy. They're trying to dig the cat out from underneath the couch. Yeah, poor cat. Don't worry about the couch. Anyway, so just a few kind of units. Talk a lot about kilobases and megabases. Really, it's a increase in factor of 3, right? So a kilobase is 1,000 nucleotides, 10 to the 3. Megabase, million, 10 to the 6. Gigabase, billion, 10 to the 9, right? So we have 3 point something gigabases, essentially, in our haploid genome. So really the only, as far as I can kind of figure out, the only somewhat correlation that you have in terms of genomes and evolutionary lineage or phylogenetic tree is the higher up the tree, supposedly you are, like us, the more chromosomes you have, right? So the actual size of your genome has no correlation to where you're at. But it seems that the number of chromosomes you have does change somewhat with organismal complexity, right? So for example, flies have 4 or 5, worms have 6, C. elegans that is, not annelids. We have 23. Chimpanzees have, I think, 1 or 2 less than us. Can't remember exactly. Right, that's about it. And even that is a not particularly robust relationship. And so really, genome size is an evolutionary kind of like hangover, right? It's like baggage. And so, as I said, with a lot of plants, the reason why they have huge genome sizes is through multiple rounds of genome duplication, right? Where somehow, at some point in their past, they doubled their genome size, their chromosome count, right? Through some error in cell division, one would presume. And the other reason is basically a huge amount of non-coding DNA, right? So essentially, or non-protein coding is actually a better way to put it. And that's really essentially the huge majority of your DNA or your genome. So in other words, most of your genome does not encode proteins. Some of it encodes non-coding genes. And that's really unknown. It's a known unknown, essentially, of non-coding genes. And so those are essentially RNAs. So ribosomal RNA, for example, is a non-coding RNA. So I'm one of the ones that I'm most familiar with, but there are more types than I know of. I know there are more types than I know of because I once looked at a list and I couldn't remember all of them. So there are lots of non-coding genes as well. But even taking into account all of that and the fact that we don't know how many there are, most of our genome consists of what could be called junk DNA. So I'm always a little cautious to use that term because it's somewhat pejorative, right? Kind of implies that that DNA has no use. Whereas really what it means is that we don't know what use that DNA has, right? Or what it does. It could well be that it does absolutely nothing. That's quite possible. Could be it does something that we no longer need because our environment or our way of life or whatever has changed. Or it could be simply that it's something that we only ever need in very extreme conditions or circumstances, right? Extreme famine, for example, or who knows what. But a bunch of our DNA, and it varies from organism to organism. This is actually one of the main reasons why genome size varies so much. Oh, my tropical fish of a wife is complaining that it's so cold. Yeah, there you go. Want to see my wife complaining? Where's she gone? There she is. Just complaining about being cold. Anything less than like 80 degrees is chilly, according to my wife. Whereas for me, anything more than 80 degrees is baking. So anyway, this stuff is called satellite DNA. So this is a bunch of repetitive sequences caused by a lot of different things. Could be caused by viruses, could be caused by transposons, right, which we'll talk about on Wednesday. Also, sometimes it's caused by errors in DNA replication, right? DNA polymerase slipping, essentially, or stuttering. That's one of the things that leads to polyQ diseases. What's good? I think ALS is one of those, or Lou Gehrig's disease. You're probably more familiar with it. And what's Q? Glutamine, I think. I think that is, let's just double check. Don't want to give you the wrong information. That didn't help. Huntington's, maybe? Yeah, I don't know. Spinocerebellar ataxia. That doesn't sound good. Oh, yeah, Huntington's. Maybe not ALS. I know that's caused by a similar kind of problem. Might not be polyQ. Actually, I had a friend of the family who died from that many, many, many moons ago. Anyway, so these consist of tandem repeats, right? So they're repeats of the same bases, kind of over and over and over again. This is quite often called satellite DNA. I don't know why. Kind of confers the idea that it doesn't have a function. It's just a multiple repeat or expansion of the same bases. And it kind of varies depending on what kind of sequence you're looking at. All right, so the big chunk of our genome is single copy sequences. So single transposons, genes, actually protein coding genes equals about, it's hard to say exactly, 3 to 5% of the human genome. That's it. Very, very little of our genome actually encodes protein, and yet we still have it. And largely, I think you can think of it in a sense as there hasn't been sufficient evolutionary cost to having all that DNA. So it's either neutral, right? In which case, it doesn't matter whether we have it or not, it doesn't make a difference in terms of our fitness, or there's some kind of fitness benefit to some of that, right? Who knows what exactly. Got a smaller chunk, which is stuff that's present in tens to hundreds of times. And then you have these highly repetitive sequences, right, which are present thousands of times in the genome. And they're typically fairly short, 5 to 300 base pairs, over and over and over and over and over again. And quite often, like the reason why I brought up poly-Q disease is that, sorry, is that when DNA polymerase hits those repetitive regions, quite often it adds extra repeats, particularly if they're very short, because DNA polymerase seems to have issues with repetitive DNA. So part of that expansion is simply a an ancestral one, essentially. There's also a huge variety in gene density within our genome as well, right? And so if you order these chromosomes by size to give them their number, you can see that they range from nearly 280 million base pairs, right? Chromosome 1, all the way down to 45 and 48 million for chromosome 21 and 22, right? So they're very hugely in size, but that size does not correlate with number of genes, right? Well, I mean, it does kind of correlate with number of genes, but it doesn't correlate very well because the gene density varies hugely, right? And so, for example, chromosome 21 and 22, right, both about the same size. 22 has twice as many, actually more than twice as many genes as chromosome 21 does, right, 787. And the density varies hugely, right? Really low on the Y chromosome, which is what you'd expect because it's a kind of stunted sex-specific chromosome. But even on chromosome 4, which is a couple of 100 million base pairs, right? A couple hundred megabases, has the same gene density as X chromosome, right? And, a third of the gene density of chromosome 22, right? So there's like the main kind of take home from all of this is that there's a huge kind of variety in terms of genome sizes between different organisms, even fairly closely related organisms. There's a huge variety in size of the chromosomes themselves, and of those, there's a big variety in terms of how many genes you have per unit of DNA, right? And really, this is all the consequence of variety of events that have taken place over the course of the evolutionary history of that particular organism, right? So we're essentially the sum of all of those changes, right? You and I, in terms of chromosome duplication or splitting or merging or, all kinds of events that have happened over the last, I don't know, like in terms of human evolutionary history, probably looking at least 500,000 years, right? If you kind of go back to our earliest common ancestors, essentially. Okay, so the other thing, the problem that this leads to, and don't forget as well, these images, or even this one, are not what your DNA look like normally. Actually, normally your DNA looks like this, right? So if you were to image individual chromosomes, which you can do, you can actually image individual areas of each chromosome and see whether or not they are transcriptionally active or not. I've spent a couple of years trying to find a video of that, but unfortunately, I think I saw it at a conference one year and I haven't been able to find it. But you can actually, in live organisms, look at an individual nucleus and see which regions of the genome are being active or not, right? So this is what it looks like normally, right? It's a ball, right? Nuclei are, spherical, and each chromosome has its kind of region or territory, right? And so these aren't neatly kind of squished up little hair scrunchy chromosomes. These are long, convoluted, complex, and even if you kind of look at it zoomed in, threads of DNA, right? So these are in, leave her alone, dog, in a very particular form, right? So the challenge is fitting all of this DNA into a nucleus, right? But still being able to access bits of the chromosome as and when you need to, right? And so this is a very dynamic situation. It's very much the case of, you have things tidied and tucked away, but still accessible, right? And that's very, very important. In fact, how accessible or not that DNA is actually a very important large-scale way of controlling gene expression, right? So you'll find that many genes that do the same, go on, up you get, process or involved in the same thing. Come on then. Go and say hello. Could someone speak? Might be able to get a head tilt out of it. Oh, no, she's cleaning the bits. Great, thanks, Dob. Just what you want to see in a genetics class, right? Anyway, where was I at? Right, so yeah, so quite often genes involved in the same process are kind of opened up or switched on or turned off en masse via the dynamic chromatin or chromosome packaging, right? And so the way that this is done is through the formation of chromatin, right? So this is, and that's essentially kind of what it looks like. So each of these threads down here, when you zoom in, right, each color equals a different chromosome. That's what this color scheme is all about. This is obviously a computer generated image, but it's generated from, actually, I don't know how they managed to generate it, but presumably they labeled individual chromosomes and figured out where they were at in a nucleus. But this is what that DNA looks like, right? So it's a multiple kind of wrapped thread of protein and DNA, right? The DNA being the gray kind of squiggles, the protein being the, I don't know, tan balls around which the DNA is wrapped, right? So this basically solves the conundrum, right, of fitting the DNA into the nucleus, but having it still be accessible, right? Well, that's actually kind of fun. If you stretched the genome from one cell from end to end, it would be 2 meters long. So about as long as I am tall, which is, well, a little bit longer even. Actually, I once heard somewhere that if you took every basically pureed up a human, presumably dead, and stretched all of their DNA out from end to end, it actually reached to the moon. I don't know if that's true or not. It's kind of a fun thing to think about. So part of that is just by twisting up the DNA, right, by supercoiling, right? So remember we talked about supercoiling ahead of the replication fork. Hey, leave yourself alone. In DNA replication, well, you can actually induce that via topoisomerases, right? So we learn about topoisomerases in terms of relieving supercoiling, but you can actually add supercoiling as well through that. But the main way is through the formation of chromatin. And chromatin is basically histones plus DNA, right? They are highly, highly conserved proteins. And what that basically means is that any mutation results in a non-functional histone protein. That's essentially what we mean by highly conserved, right? It means that all of those proteins, all those amino acid residues are important. And so change in any of them typically results in a non-functional protein, which is not good. And these basically form what are called nucleosomes. All right, so there's a little bit of language going on here. So we've got a nucleosome, right, which is basically the amount of DNA from the beginning of one core particle to the beginning of the next one, right? And so that's basically around 200 base pairs, give or take. I'll type this out. And that's the DNA from start of one bead, I guess, to the start of the next. And that includes DNA wrapped around one bead and between the two beads. So one nucleosome core particle equals just the DNA wrapped around the histones, not between the beads. And that equals about, 145 to 147 base pairs. So essentially there's around 50 base pairs of DNA between each bead. That's the stuff that you can see at the top. And the reason for that is that it allows these, as you can see here, these beads to be wrapped around each other, right, to create that higher order structure. It also, importantly, when we come to gene expression or control of, it allows these beads to be moved up and down the DNA, right, rotated or slid, right, like in an abacus, essentially, right, to allow some DNA to be more accessible or exposed than other parts. And when we look into it in more detail, again, this is, I'm pretty sure I've shown you this already. I think this in another PowerPoint, right? Each nucleosome core particle equals 8 core histones. Was that H2, 3, and 4? Oh, 2. Essentially 4 pairs is an easier way of typing it. 1H1 protein. Holds stuff together, basically. And around 145 to 7 base pairs DNA. And these histones, particularly the 8, H2A, B, 3, and 4, right, 2 pairs, these are really important because they have properties that can be modified, right? So histones are actually, let's see if we can remember this. What charge do you think histones would have to make DNA stick to them? Questions to the class, so it's not just me chuntering on. Yes, awesome. Thank you, everybody. Positive. Why? What makes DNA negative? Anybody remember? That's more its structure, not the reason for its charge. No, it's not the hydroxyl, because the hydroxyls essentially don't exist. It's not the phosphodiester bond. Phosphor something. So what two repeating units make up the backbone of DNA? There's the sugar, the ribose. What's the other one? It's the phosphate, exactly. And phosphates are negatively charged. Actually, I was a little bit too hasty. It was Caitlin said about the hydroxyl. It's actually the hydroxyl group on the phosphate. I think you may have been thinking about the one on the sugar, which doesn't exist because it's turned into a phosphodiester bond. It's the OH, which becomes a negative O on the phosphate group, is what gives DNA its negative charge. So histones are positive. Now, you can't change the properties of DNA because it's a pretty static molecule, but you can change the properties of histones, right? And so we can change how tightly these proteins attract DNA between lots and none, essentially, right? So we can actually dynamically change the relationship between DNA and proteins in chromatin. So just a wee bit to finish up with. We already talked about this, but talking about it in a kind of slightly different context. It's actually kind of weird to think about, but if you think about the nucleus as a ball, right? It's not a solid ball, right? Even though it has stuff in it. If you think about it as a, I don't know, a soccer ball, let's say, right? Or a volleyball, whatever. Essentially, you have a membrane, right? The nuclear membrane, right? Kind of the encapsulates the nucleus, is what makes the nucleus what it is. Now, in that, the chromosomes are actually attached, right, at a variety of points to the inside of that membrane, right? That's what gives them their territories. So if you think about each kind of, basically each chromosome is going to be sort of like stuck like so, and they're all kind of stuck around the periphery of the inside of the nucleus, which is kind of weird to think about, really. You know, it's like one of those fractal patterns. You know, the more you look at this stuff, the more there is to look at, and the more there is to look at. It's just astonishing, I think. I wish I could find it. Let's see if I've got a few minutes. How would I put this? Oh, if I could spell it right. Eh, trying to see if I can find some good pictures. It's a bit of a challenge sometimes. Well, this is not quite what I'm looking at. Hello, what's this look like? Oh, that's during mitosis. Not interested in that. Nah. I've spent a lot of time actually. Ooh. Nope, that's not what I'm after. Yeah, look, lots of pretty pictures, but not really what I'm after. Actually, this is, I think, probably a better one. I think this must be Drosophila, would be my guess. Yeah, in fact, it is definitely Drosophila. These might be salivary gland cells. Oh yeah, there you go, polytene chromosomes. This is actually even weirder. So in, let's see if I can get this, invigorate this. So in some insects like Drosophila and their salivary glands, they undergo multiple rounds of DNA division without undergoing cell division. And so this is a, what are they using for the visualization? So these are using different GFP reporters, so fluorescent protein reporters. And so this is in one nucleus. You can actually see these, they're really weird chromosomes because they're kind of like very, very wide, puffy. But you can actually see, and there's different regions which glow. Those are the regions that are being transcriptionally active, right? So for example, this region here and the brighter bands in these other regions, right? So that's one example. You can just kind of tool around the internet and see if you can find others. The ones that I was thinking of use different fluorophores, right? So ones which I forgot the right word, brain fart, glow in different wavelengths. And so you can actually look at different regions simply by using a different excitation wavelength for the different fluorophores, which is pretty cool. So yeah, there's like the limits of microscopy are getting ever, higher and higher in terms of what you can actually do. It's really quite astonishing. So essentially this DNA packaging kind of goes between multiple different levels, right? So essentially what we really talk about or care about is difference between euchromatin, which is open, transcriptionally active, and typically the 30 nanometer structure, which is what we're looking at here, versus heterochromatin, which is closed, transcriptionally silenced, and the 30 to 300 nanometer structure. So you can have heterochromatin simply by making it transcriptionally inactive. But part of doing that is kind of folding that DNA kind of even more around itself to create this thicker structure. Now, these ones down here, these really wide fibers, those you only see during cell division, mitosis and meiosis after prophase. And so the length actually reduces by, a factor of about 10,000, right? Which is quite astonishing. And it has to do that because you want to, even in mitosis, you want to segregate your chromosomes faithfully. It's even more important in meiosis because of the importance of recombination as well. And just to kind of, I think we talked about this in terms of map distance versus physical distance. But here's another way of looking at it as well, right? So the arms are typically euchromatin, not always, right? Because it varies. It's a dynamic state between open and closed. But the center of the chromosome, wherever that might be, it's not actually always in the center, but the centromere and the telomeres are typically heterochromatin, right? Highly coiled, very few genes, right? Transcriptionally inactive, an awful lot of satellite DNA as well, right? So even just going along a chromosome, not just between chromosome and chromosome, but in different areas of a chromosome, you have a huge difference in terms of gene expression and the number of genes there as well, right? So it's a very complex picture. It's not a uniform deal. And that's pretty much it for me. So I'm gonna sign off. I have a trip to a scrapyard with my buddy, which will be super fun. And I'll upload all this stuff when I get back, when it's finished processing. So have a lovely day. It's a beautiful day out there. Great for yomping around a scrapyard. And I shall see you either tomorrow or on Wednesday. So have a lovely day and see you soon.